Welcome to this explainer. Today, we're looking at how forensic scientists are taking degrading genetic material and turning it into a highly precise microscopic molecular clock to estimate the exact time of death. So let's jump right into it.
In forensic investigations, traditional methods, like checking body temperature or digestion, they get really unreliable during the first 48 hours. But the solution? It's been hiding in the predictable degradation rates of RNA. By carefully mapping that degradation, the body essentially reports its own exact time of death.
The researchers put together a massive, highly integrated workflow. First, they use high-throughput RNA sequencing, basically casting a giant net over brain tissue across different time intervals after death. Then they use differential expression analysis to sift through that mountain of data and find which specific RNA transcripts are changing over time. Next, they take those findings and rigorously validate them using real-time quantitative PCR, pairing them up with incredibly stable reference genes. And finally, they feed it all into complex statistical regression models to translate those microscopic signals into a precise timeline.
When you look at the data, messenger RNAs, or mRNAs, predominantly degrade over time, which is probably what you'd expect. But circular RNAs, or circRNAs, they show a completely different story. A huge proportion of these actually show a relative upward trend. Out of thousands of differentially expressed circRNAs, over 58% were actually upregulated, meaning they stabilize long after the organism dies.
Think of it like this. Messenger RNAs are linear. They have exposed ends, which makes them highly vulnerable to what's called exonucleolytic decay. Basically, that's just cellular enzymes coming in and chewing them up from the outside in. But circular RNAs, they form a covalently closed loop. No exposed ends at all. Because of that closed loop, they are incredibly resistant to this decay. So while the linear mRNAs are actively breaking down, the circRNAs hold the line. This makes them incredibly potent, highly stable biomarkers for forensic analysis.
To make sure their PCR validation was scientifically airtight, the team rigorously screened for stable internal control genes using evaluation software like RefFinder and Genorm. And they didn't mess around, they applied super strict mathematical thresholds. We're talking an average expression stability, the M value, strictly below 1.2, and a pairwise variation, the V value, below 0.15. By running the data through this intense filter, they identified rock-solid reference markers like 5S, RPL13A, and MIR125B.
When you map the stability of these markers, the top performers, like 5S and RPL13A, literally separate themselves from the pack. They hit those impressively low M values, proving they are vastly more stable than any of the alternatives. By using these optimally stable markers to normalize the data, the researchers can be absolutely sure that any changes they see in their target RNAs are caused by the actual postmortem interval and not just biological noise or experimental errors.
So we have our validated markers and our stable baselines. Now it's time to model the data. Enter PCA, or Principal Component Analysis. It cleanly takes these chaotic molecular markers and separates them into clear, distinct, time-based clusters. We're talking a perfect progression from 0 hours, stepping neatly all the way down to 48 hours. The data naturally organizes itself. This proves beyond a doubt that the chosen RNA markers carry a powerful inherent signature that corresponds directly with the time of death.
But to get the absolute most accurate quantitative prediction possible, the team applied partial least squares regression, or PLSR. Now, their original 17-marker model was good. It had a solid prediction R-squared of 0.815. But by zeroing in on the markers with the absolute highest impact, they optimized the whole model, cutting the number of required markers down from 17 to just 5. The result of that? The prediction accuracy actually jumped up to 0.859, and the error rate, the RMSEP, dropped significantly, going from about 8.9 down to 7.0.
So, by trimming the fat, they reduced those 17 initial candidates down to just 5 incredibly potent core markers: S100B, LNC-TUG1, ARP19, TNC2, and beta-actin.
When you compare the models against measured time intervals, the optimized 5-marker model tightly hugs the ideal prediction line, especially at those tricky 12 and 36-hour marks where things usually get messy. The test data falls much closer to the expected timeline than it did in the unoptimized model. This allows for a highly reliable, truly quantitative calculation of the death interval, proving once and for all that carefully prioritizing the right markers absolutely outweighs just having a sheer quantity of them.
Which leaves us with a pretty thrilling prospect. We are literally looking at the future of forensics.